Hello and welcome. Today I'm going to talk about a lady, Henrietta Lacks. She was an African American black lady. She passed away in 1951 at Johns Hopkins in Baltimore in a hospital. But the remarkable truth about this lady is that she's still alive. If we don't go on to define what is life. The reason is something that I'm going to discuss about today. So, this lady got an very aggressive cervical cancer, and cancer is uncontrolled growth of your cells. So if you take any cells of your body, so they keep on growing, they divide, but after a certain time, let's say after 50 division, they won't divide, but they go for a programmed cell death. But if you think about a cancerous cells, generally cancerous cells keep on dividing, and the reason for this cell proliferation, which is not controlled in case of cancer, in order to understand that, we need to understand what makes a cell to divide even in a normal condition. So normal cells, everything that happens in a cell, inside the cell, that is being done by different proteins, and these proteins are, I always call it, molecular worker inside the cell. They work. And what does work essentially mean? That they bring some changes against the already existing force. So and that work from physics. So the protein machinery, and inside the cell we have millions of protein, different kind of proteins performing different kind of work. So, and how does protein form in the cell? Protein forms in the cell by another machinery, which itself is made up of protein and some RNA. We call it ribosome machinery, and there protein synthesis happens. And when and which kind of protein will form, that depends on the expression of gene. So inside the cell we have a storage house for this information. That information, we call the information, is coded in DNA, and this DNA resides inside your nucleus, and a part of DNA that has code to make messenger RNA and protein, we call them gene. So there are different genes inside the cell. So in order to discuss this story about this remarkable lady who is still alive even after her official death, medical death, I should be discussing with you three kinds of genes. So I should be saying three sets of genes. So three sets. So when I say gene will form some sort of protein. So I'll talk at protein level only. So what happens? There are genes which are called growth promoting genes, and these growth promoting genes inside your nucleus, inside any cell, has codes for proteins that somehow promote the cell proliferation. And how do they do this? Let's talk about a growth factor, let's say a growth hormone. So what happens? Growth hormone secretes from your pituitary, and let's say it comes down in your blood, and on any cell, let's say on a kidney cell, it just sits on your kidney cells, and kidney cell has a receptor we call growth hormone receptor. And when growth hormone receptor interacts with the growth hormone, then and this receptor, growth hormone receptor, is is a protein which is present on the membrane of your kidney cell or any cell. And further, there are next protein which sits inside the cell, and that protein gets activated when growth hormone binds to the growth hormone receptor. And this protein again activates another protein. When I say activate essentially means that they interact, they changes their conformation. Sometimes they change the conformation of protein such that it becomes it takes a shape that can interact with another protein, and then the another protein. So this is how a growth hormone comes, sits on your cell, any cell. It binds with your growth hormone receptor. It causes further changes inside your cell at multiple protein levels, and then it goes and activate the protein that is involved in cell division, and further cell division happens. So you have to name this protein as an all set of protein who are involved in this cell division process. We call them growth promoting proteins, which is synthesized by us. We name it as a growth promoting genes. Now the important point is our nomenclature. Protein molecules, the genes have naturally evolved million of years ago to function this way. Just for the sake of understanding, we name it. Having said that, another kind of protein molecules, and these protein molecules are involved in suppressing the growth. So let's say cell divided, not divided. After attainment of a certain, let's say your organ has taken a certain shape, or maybe there is a cut, there is a wound, and naturally immune system would like to fill the wound, and in order to fill the wound you need to synthesize some cells. You have to proliferate cells in that area. But after healing is done, you would not want your cells to go on proliferating forever. So there are a set of genes who forms a set of protein, and we call them tumor suppressor gene, or cell growth suppressor gene, who makes cell growth suppressor protein. And they do not want your cell to go on proliferating forever. And there are third kind of gene, we call them apoptotic gene. And these apoptotic genes are the genes who make certain kind of proteins. We call them apoptotic proteins, and these apoptotic proteins kill the cell. But this killing is not because of some enemy purpose, but because of goodness of your health, goodness of your function of the system. So if a cell has proliferated enough, let's say 1500 times, or maybe a certain number of time it has already divided. After that, what happens if your cell keep on proliferating for long? There are chances of multiple mismanagement at the cellular level, at the DNA level. So what happens that if your cells keep on proliferating, maybe DNA will mutate or something will happen. So it's natural that cells go through, after a certain division cycle, cells go for a programmed cell death, and this programmed cell death we call apoptosis. Again, it's just a nomenclature. Nature, evolution, cell system, living system, physical laws, they don't work as per our nomenclature or as per our books. Rather, they work in their own way. We read them, naming them with some, if whoever leads, they get a chance to give the nomenclature. So presently in science, English people lead, and they give this kind of English names to it. if you go in spiritual science, Sanskrit people leads, and they have given a lot of terms and ways to seek the 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 super or maybe higher knowledge, we call it. So they are named in Sanskrit. Okay. Having said that, so what I am trying to make you understand that how a cells become cancerous. As I said, cancerous cells are the cells who keep on dividing without a stop, and they can keep on dividing and make a tumor, and that tumor can be harmful for you. So, but normal cells do not do that. Normal cells grow when they need, when they don't need, they don't grow, and also they go for a natural programmed cell death after a certain time and new cells form. Now the question is, why does this phenomena happen that we call cancer? So if your tumor or growth promoting genes, if they become super activated, if they get super active, they will make a lot of growth promoting proteins, and those growth promoting proteins will keep on facilitating cells to keep on dividing and dividing. This is one reason. Another reason is if tumor suppressor genes become suppressed, then they would not make enough tumor suppressor protein to suppress the cell growth, and in this scenario also there will be unchecked multiple division of cells, and it will form the cancerous cells. And third reason, if your apoptotic genes which lead a cell to go through a programmed cell death, if they don't work, then also your cells can go on proliferating forever, and it will again make the cancerous cells. Today, after defining what is cancerous cell, I'm talking about the lady, and that lady got a very aggressive cancer, cervical cancer, and she visited a hospital at Johns Hopkins in Baltimore, and there people found that her cells are way different than even other cancerous cells. Of course, it was cancerous cells, and definitely it has to be different than normal cells, but perhaps those cells have multiple mutations. So one thing that question you should be asking, that why do tumor suppressor proteins or apoptotic proteins or growth promoting proteins, they starts behaving differently? Because of some mutation in them. And if what is mutation? Mutation is simply change in amino acid. If so, how does it matter? Like, what is the sequence of amino acid? If let's say your valine is replaced by lysine, what would happen? So whenever an amino acid, so these sequence of amino acids decides the structure of your protein, the stability of your protein, the dynamics of your protein. So everything is linked. How is it related? We can talk about that later, because of some chemistry, because of some interactions. Having said that, so because of mutation, these proteins and these genes can alter their function, and when function gets altered, there will be alteration at the emerging property level. So if protein gets altered, get mutated, then if let's say if your tumor suppressor protein has got a mutation that lost its function, then it will be unable to check the growth of your cells. And if there will be a gain of function mutation in growth promoting proteins, then there will be again uncontrolled cell growth, because these cell growth promoting proteins have gained an enormous power of function to not dying. Proteins have also a limited time inside the cell. They form, they do their function, and then they vanish. Vanish means they just break down into different amino acids. Then again, whenever which kind of protein is needed, that is decided by certain other proteins, and they go and talk with your DNA, and then again they give some protein. But at the bottom level, every protein has a life cycle. Like after doing this work, they will just break down into amino acids, and those same amino acids will be collected at the ribosome to form another set of protein. Now, if there is a gain of function mutation in a growth promoting genes, growth promoting proteins, then the protein will not die easily. This is one of the reasons that they will remain stable for a longer period of time, and they keep on working, they keep on proliferating the cells. Now, when I say mutation in protein, that one amino acid gets replaced by another amino acid, then it's important to understand that mutation happens at the genetic level. So if there is a change in DNA, then there will be change in messenger RNA, which is transcribed from the DNA. And if there is a change in messenger RNA, same will be translated into the protein. Sometimes mutation happens in the messenger RNA as well, and multiple things can happen. Just to confine this discussion to about the lady, so she got this cervical cancer, and the problem was her cancer was way different than the normal cancers. Even in cancerous cells, there is, they will proliferate for long, but after certain proliferation, they would like to stop. They don't go on forever. But these cells, they found these are sticky cells at Johns Hopkins doctor. They found that these are sticky cells, and they keep on proliferating forever. Of course, when they get nutrients, nutrients are needed to proliferate a cell for a cell division. And this is a great example where you see that some organic and inorganic molecules, which we called non-living, those non-living molecules, organic or inorganic molecules, they are taken by the cells, and then your cells use those molecules to make 2 out of 1, and from 2 to 4, 4 to 8. So essentially, when a cell divide, they do not develop their components of cells by their own. They just take it from nutrients. For us, what we eat, those food gets absorbed in our blood. From blood, they go in different kind of cells, and in the cell, they are getting used by different machinery, protein machinery, to form another required things. Maybe some protein, maybe some organelles, maybe some plasma membrane, some different sort of things that exist in the cell. So cells form from an inorganic or an organic so-called non-living matter. So this is a beautiful example where non-living thing is converted into a living thing. Okay, I'm talking about that lady and what happened. So, when her cells were put in a media, her cells keep on proliferating forever. Like there is a no death of those cells. And then people thought that, wow, this is something very interesting. Like earlier, before 1951, every scientists were using cells to study different kind of questions, and the problem was that after 40-50 division, cells would die by the process of apoptosis and so on. But these cells were remarkable, were not dying even after, let's say, 1000 division, and this was amazing. If you ask, it's still controversial that why does it happen. There are logical arguments. I will not go into that detail, but I'll tell you that there are some mutations exist in these cells of the lady, and those mutations perhaps somehow function in a way that cells never die, they keep on proliferating. I work on those cells, and those cells are still alive, proliferating, starting from 1951, and it's 2021. Cells are still alive, cells are still proliferating, and today I am going to show you that cells. We call them HeLa cell. I can say that lady is still alive, and I owe you at least a thanks to her for this beautiful contribution. Although she passed away in 1951, she was a mother of 5 children. She faced a lot of racism because of her color, and those times were the time when this racist mindset was at the top. I felt really bad when I read that if color is black, then that black colored population was serving as slave. So there was slavery based on color. In India, we witnessed slavery based on caste. This is not good. This is not good for the people. This is, of course, not good for the slave, but this is not good for the master as well. And here there is an ethical point that comes up, that these cells came into research. Scientists started using these cells even without her consent, even without consent of her family. Even today, she, as I told you that she had 5 children, nobody, not a single scientist went and got consent of even her grandson or maybe great grandson. Nobody asked them. Of course, we cannot go back in past and revert the action, and we ask for consent, but acknowledging the fact that this was wrong, using somebody's cell without her consent is unethical. The acknowledging this fact would be far way better than saying sorry. But I have my deepest condolences, and of course, I thank the lady for this cells, because I work on that cells. So this is called HeLa cells, and today I'll show you how does it function. So first of all, what happens that uh, basically we have cells, and cells we grow in some nutrients. As I said, that we always grow cells in 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 the presence of some nutrients, otherwise they won't survive. Let's say if you don't eat, okay, then definitely you will not get things that are needed to maintain the healthy life of cell as well as to proliferate. Hence, we keep these cells in some media, and let me show you how do we do that. So here it is. I am literally holding Henrietta Lacks cells who passed away in 1951 today. So she is in my hand, all alive. I am going to show you. So as I said that we grow cells in some nutrient media. You can see that this red color thing is a media. We call it DMEM media. This media has got everything like glucose, like amino acids, like food supplement, like growth factor, like different kind of growth hormones, everything it has got which are needed to maintain and proliferate a cells. So we have cells in that media. Can you see the cells? Of course no. How would you see a cells which has got a size of 20 micrometer in 1 and 10 micrometer in its width? It's extended cells. So in order to see these cells, to visualize these cells, we need something that can magnify things. Okay, magnification means, so this our eye has a limit of resolution, limit that you can distinguish two things to a limit. If something is very small, you cannot see them, and that's the limitation of our eye. But a good limitation, just assume that when an electromagnetic radiations come up here, like there's a white light coming here, which is an electromagnetic radiations, and let's say during the daytime you're walking and you see something in sunlight. So when you see only 7 color as per our categorization of this visible spectrum that we called violet, indigo, blue, green, yellow, orange and red. So this is the spectrum you see. Assume that you get an ability to see even wider spectrum of electromagnetic radiation. Let's say you cover infrared also, you cover X-ray also. So what will happen? So when electromagnetic radiation from sun would fall on an object, and they would reflect X-ray and gamma ray and infrared rays as well, if your brain would be able to detect these radiations as well, then an object would look like pattern of electrons. Like let's say that this, if you have an ability to detect X-ray that are getting reflected from this object, then you would not see this object like this. You will see an object completely different, like a pattern of electrons or protons or atoms, and that would be really bad. So today what we are is a result of natural selection. We are at its best version. Hence these words like limitations should not be taken in a negative sense. Whatever we have that for a good reason. Let's concentrate about the lady, and she is waiting there, and I'm going to show you in microscope that how can you literally observe the lady and their cells and acknowledge that. Okay, so here's the microscope. I'm going to show you how do we use the microscope, and the picture I'll take of these cells, I'll just put it in the video in some link. Okay, so here I am on the microscope. So this is the cell, okay, that I'll keep on a surface, okay, And this is the eyepiece. Eyepiece is something that I can with which I can look at the cells. Right now I am looking at the cells. So these HeLa cells, you have to trust me that I am looking at the cells, because one of my friend Nikhil, he has this remarkable ability to capture cells through eyepiece in a camera, but I am not that skilled. But I can use a laptop to capture that. But just to explain what is a microscope. So microscope is an instrument, okay, made by some engineer, and it has some lens. So what does lens do? Lens just magnify things, okay, and it has certain other things to get you the resolved image, better resolution. So there are different objective here that you can rotate 10x, 20x, 40x, 63x, 100x, and so on. X means how much you want to magnify something, and that is how you can use. So presently, if I want to use, let's say 10x, then I will take it as a 10x and I'll look at the cells. Second, here, from here, if you can see, from here there is a source of light. You can see I can just increase or decrease this intensity of light. See, here you see the intensity of light is increased. If I reduce it, intensity of light is decreased. So this is the source of light. So right now there is a light in the room, but if you don't want that light, then definitely this is the source of light. And of course this room light cannot be used to just look at the cells. Okay, so we have cells, and these cells are nicely spreaded throughout everywhere, okay, but these cells are about 80% confluent. When I say confluent, I essentially means that when I say confluent, then I essentially mean that so cells are nicely spreaded on this surface, okay, that I visualize through the microscope. Now what happens, and I'll put a picture just for the evidence that there are cells here. Now what happens that there is a limit. As I said, that these cells can proliferate forever, but there is a limit. Like if I ask you that in a room, let's say there are one person, of course they can keep on living in that room if you can keep on providing them food. Now if there are 2 persons, that is also fine. 3, 4, 5, 6, 7, 8, problem. 9, 10, problem. And there will be the same supply of food everywhere, and there will be struggle for food. And then if it goes like if it is a normal our house room, and if number of people are more than 20, then you cannot serve, like more than 20 people cannot survive in a room. And you see that I am doing this growing these cells in a closed container. It does not get anything from outside. Even when I'm speaking, there are multiple virus bacteria that are coming from my mouth, but it cannot enter here. Okay, now and we grow it in a closed environment. So there is a limit to which it will proliferate. After that, there will be competition among cells for nutrients. There will be contact inhibition. Like if you are nearby multiple cells, then definitely you will face some kind of difficulty. And hence, now I just looked into the microscope, and I found that about 80% of this area is covered by cells. Now if I leave it, it will cover 100%, and after 100% cells will start dying, not because of apoptosis, but because of the competition for food, because of contact inhibition. Like one cell will like to inhibit the growth of another cell. That's how nature works. But we should not be doing that in our day-to-day life, because in your room you are only 1 person or maybe 2. So intense competition is not needed. Live, that is important. And also for you, are free to go wherever you want. Here I have closed it in the cell. I don't know how much punishable offense it is. Maybe God will punish me for doing this to the the lady who is just in my hand. Okay. So what should we do if it is already covered by 100% and by tomorrow it will spread in like in whole area and there will be death of cells? I don't want that. So what I'll do, I'll just split the cells. I'll take out, like it has let's say million of cells or maybe 10 million of cells. So I'll take it out. I'll just fragment these 10 million cells into 10 different vessels. Or maybe if I don't need 10 million cells, then I'll take 1 million cells from it and I will grow it in another vessel where I can again maintain the cells and use it whenever I need it. Okay, so I don't grow these cells and use it just for my fun. I need these cells. Why? Because for me, I am a physical biochemist. For me, I just want to understand some thermodynamics of the cell. But for other people, there are multiple regions. They want to do some experiments in this. And hence, they don't want to kill these cells by contact inhibition or any other process. So what I'll do, I'll split these cells. Generally, in scientific language, again, language is just a language. Nomenclature is just a nomenclature. Things don't work as per nomenclature or language. Things work as per laws of nature. So okay, so there is a term, nomenclature, for splitting these cells. We call passage. Like we passage the cells, or maybe we call it subculture. Like this is cultured cells, and now I will subculture into multiple wells. And in order to show that how does that happen, I need to show you certain things. So first I will keep these cells in a safety cabinet that I'll show you in a minute, and then I'll show you how to do that. First I'll show you the theory part, and then I will make you understand how things work in an experiment. Okay, so in order to in order to get it done, the subculture, first of all I need fresh new this flask, okay, in which we'll grow the cells. Second thing, we need the media. Media is the growth media. As I said, media has different things like glucose, fructose, all nutrients. Why do cells need glucose, fructose and all? Because they break these molecules into smaller things like CO2 and H2O, and then when they break these things up, means things are going from ordered state to disordered state. And whenever things go from ordered state to disordered state, it's a although it's a spontaneous process, but spontaneous process means what? Going from high energy to low energy. So when it is so if you look at the structure of glucose molecule, and so glucose molecule has high energy stored in their bones. So low entropy means high energy. Okay, so anyhow, so when you break down the glucose molecules in respiration reaction, then you are essentially increasing the entropy, and when you are increasing the entropy, you are gaining the energy. And what is energy? Energy is capacity to do work, and cells do work. Inside the cells, protein molecules do work, and in order to do those work, you need energy. And here we give that energy from outside source. How do we get energy to do for proper working of the protein molecules inside the cell? From food. In the same way, so I am actually taking care of the lady by giving her food. I give her glucose. I give her different amino acids. I give her supplements, growth supplements, different kind of serum factors, and a lot of things that are needed for their growth. Apart from that, I also add one thing in that, some antimycotic and antibacterial compounds. So antibacterial compounds are penicillins, and antimicrobial compounds are something I just forget the name. That is fine. Name is just a name. It's a compound that inhibits the growth of fungus. And penicillin, which is an antibacterial thing, it inhibits the growth of bacteria. So why? Just to take care of the lady that other bacteria and fungus, they don't attack and disturb the lady. I am the caretaker of the lady. Okay, so uh, having said that, there's one more point that I want to make here, and I don't really know what that point is. I'm just thinking. Yes, I wanted to show you the how does a media look like. Okay, and I'll show you the media. So, so this is the media. Okay, this is just some compounds dissolved in water. Okay, but this is very sterilized. Sterilized means it has no bacteria and all. It has this antimycotic, antibacterial agent as well, and it is stored in a flask with which is so much tightly bound that bacteria, fungus, they cannot enter. But mycoplasma is our enemy. Mycoplasma is the smallest tiny organ cells. We shouldn't call it cells. They are joker of plant kingdom. They do not have a cell wall. They can take any shape, and they are smaller, so less than 0.2 micrometer in size, and they can enter sometimes. And when they enter, what do we do? So I remember a Pakistani player who said that with playing, so captain Pakistani team was playing cricket with Indian team, and they, of course, they lost the match. And one time Pakistan did not lose the match, and I was very sad. But now I don't. But still, I am just. It feels good when India wins. Okay, so some media person asked him that how do you feel? What is your planning for today's match? Or maybe I don't know whether it is before the match or after the match. So he said that, see, my bowling is weak, my fielding is weak, my batting is also weak, and we have eaten a lot of food last night, so we are lazy also. So we do nothing. We just do things. So in the same way, what we do to stop mycoplasma, we do nothing. If mycoplasma enters, we just throw it away, and we have to clean the room and everything. Now, one more thing I want to show you. This another thing is called trypsin. This is trypsin, or a different variant of trypsin we call Tripoli Express. Some fancy name, but essentially this is trypsin. Now there are two things that is very important. First, when I said that I told you that I'll show you the cells, I will show you the cells. Don't worry. But believe me, believe my words presently, that in this flask, cells are adhered to the surface. They are adhered to the surface, stick to the surface. Now, when I told you that these microscopic cells who are not even visible by our eyes, how can we take them out and split into different flasks? And put them in this media. So the first thing that we need to do is we need to dissociate these cells. We need to dissociate these cells from this substratum, from this surface. And after dissociating the cells, we need to split it into different flasks. And in order to dissociate the cells from the substratum, we need to understand that how is it associated at the first place. So what happens, the cells have different appendages, and using that appendages, it just stick to the surface. Since it is an adherent cells, so it will stick to the surface. But in my case, this surface is treated with a protein called fibronectin. Fibronectin is a protein which helps cells to adhere with the surface. So this is a protein, and it has different ends. One ends bind with the cell, and another ends bind with the surface, which is already, if your flask is treated, then that will remain bind to the surface, and hence your cells will remain sticky to the surface, right? Okay. Hence, so this is how cells remain sticky to the surface. and this is how they associate with the surface, the substratum. Now, in order to dissociate, what we need to do, we need to cut off these proteins involved in joining cells to the surface. So what we are going to do, that we are going to cut off the protein involved in adhering the cells to the surface. And who will help in that? This trypsin thing. This is trypsin. Trypsin is an enzyme, and these enzymes cut the protein at, if I'm not wrong, then arginine, or there's one or amino acid. So if your protein and it has arginine, trypsin will cut the protein at the arginine level. If it is not surrounded by proline, because proline gives you some hinge where it cannot work. But at arginine, and there's one, I guess, serine. It's a serine protease. So I think it's serine and arginine. At these two places, it will chop off the protein. So here, what we want to chop off the protein which are involved in adhering these cells to the surface, to the substratum. Okay. Having said that, now you should be asking a question that when you put trypsin, then can't it chop off the protein which is present inside the cell? Can't it chop off the protein which are useful protein for the cell? Yes, it can. That's why we have standardized a protocol in which 2 minutes are sufficient enough to chop off the protein which is involved in adhering these cells. If you leave it more than 2 minutes, it will definitely destroy the cells and affect the cells, and it can chop off some important proteins for the cell. Then what will happen? Cells will die immediately? No. Cells have an ability to make their own. They are not like me and you. If you give them this, they will make it protein from this liquid, these compounds. Remarkable machinery, I always try to restrict myself not to go in things that is not meant for the video, but how remarkable it is. Like this machine, the cell machine, that it just takes this thing that I just made in the lab, I just made in my biochemistry lab, and now I am in the cell culture lab, and this thing will be converted into a cell. It will become cell. Like you put 1 million cells in that, or maybe you put 5 cells in that. You put 1 cell in this, and this 1 cell become 2, 2 to 4, 4 to 8, 8 to 16, and tomorrow you will have million of cells from 1 cells. Maybe the day after tomorrow. So essentially, I am saying that this thing is getting converted into a live cell. So how good this machinery is. So a cell, this cell, when it will become a cell, when this will become a cell, it will again use all these characteristic of life. Now the important question is is this really life? Like if there is a cell, can we call it life, or life is something else? Maybe cell is just a machinery, a big machinery. Different, an atom is also a machine where there is a proton and electron are surrounding it. When 2 atoms combine, like O and make O2, it's a molecule, and there is a compound. There's a compound like histidine, arginine, and these amino acids. Your ATGC, adenine, thymine, guanine, cytosine. These are also different compounds, and these compounds again associate and form DNA. They form protein, and these DNA and protein associate and form some simple organelle, and these organelle again interact with each other for some function, and they start doing work. Like if you talk about mitochondria, they do remarkable work of breaking down glucose into smaller molecules that we call CO2 and H2O, and gaining this entropy gives you energy to perform the various function. So these are different machinery, and then different organelles mix and form your cell, and different cells again interact with each other form tissue, from tissue different tissue forms, and they become organs and organ system, and then your body. So when this is important, that what is life, when we call that a cell is having this much property that they can grow, they can do protein synthesis, they can do oxidation of glucose, they can just make some protein, some hormones and secrete it through Golgi apparatus in a vesicle, they can store some genetic material. We are talking about property. Atom also has a property. Then why should we call atom a non-living thing? So this is something very important question in science, to define animate and inanimate in a proper way. So I just don't understand what makes O2 molecule non-living, and what makes an arginine living, and what makes arginine, lysine, glycine, histidine, cytosine, and all cysteine, all these combine and become a protein, and then these protein molecules combine and form a cell, and then we start calling cell alive. What different it is? Like if there are multiple, let's say this microscope, or maybe this mobile phone, it is also is made up of different components of it, which is again made up of atoms and molecules and compounds and plastics and iron and whatnot, and glass and everything, and they just work in an interaction. They interact with each other and form an instrument that gives you a property. Let's say a fan in your room. So fan is also giving you some property. So should we really call an emergent property as a remarkable feature of life? Because every smaller compounds can interact and they can give you a property of life, an emergent property. Like here, tomorrow I'll show you that this media will become a cell. Now, what makes it non-living in this flask, and what makes it living when it is inside the cell? I don't know. Maybe you should be knowing more. If you know, then definitely please let me know. So I made this video for kids at my home, and some junior friends who just told me to keep making these videos, just to talk, just to discuss. And this is a pandemic time, so I don't know who are watching it, but if you are watching it, definitely you should help me to understand what indeed life is. Okay. Now, so I explained that using this compound, I will dissociate cells from the substratum, and after that I will put it into different system, and then I will use them. Okay. So let's go to the cabinet where I will do this process. Okay, so here's the cells. Okay, and I'll show you how to split it. So I will split it into 4 different flasks. So I have intentionally chosen a smaller flask because I need to harvest the cells in 2 days only. So I'll show you how do we do this. So first of all, we need to fill some media in these kind of flask, and media is a liquid, so you need an instrument to take out the media, which can measure the volume, because liquid thing you measure in volume. So this is called pipette, serological pipette, and using this gun I will just take this serological pipette out. If you can see, so this is serological pipette, and then this is the media I'll take. Okay, so very tightly bound just to avoid mycoplasma. And what I'll do, I'll take media in this pipette. So I have taken around 15 ml of media. Okay, and I'll put it in this different flask. So okay, so first thing is to put in media in different flasks. in the middle I thought to stop the video and do the work and then record the video again, but then it becomes very difficult. I need to just, if I delete and if I stop in the middle and re-record it, then in order to mix them I need to invest time using some editing. I don't want that. I just want to record and I'll upload it as it is. So the important point is there is very scarcity of time. But, well, it is not entirely true. Okay, so I have filled media, new fresh media in all these flasks, and what I'll do, I'll first remove the media which is already present in this flask. Okay, so you can ask that when you remove media, then wouldn't cells also come out? No, it would not, because cells are adhered to the substrate. Okay, now I need to just throw out this media, and this media is of course there will be cells which are not adhered with it, so we'll just store it in another flask just to throw them out. Okay, so once I have taken out the old media from this, I will first do one thing that we just wash it with GTBS or PBS buffer. So what is PBS buffer? PBS buffer is this is your buffer. Buffer is something that resists the change. Okay, and this is the buffer which resists the change in pH. Okay, so it has this is a solution. Solution means something dissolved in your water, solute dissolved in some solvent. Here water is the solvent, and solutes are sodium phosphate, potassium phosphate, NaCl, and KCl and all. Now you ask me, why do I put all these things? First of all, why is there a need to wash them? So there are multiple dead cells in it, and those cells should be washed. Now why can't we just wash it with water? Because as you know, water has low osmolarity. If you put a cells in water, then water will go from its higher concentration to lower concentration, and cells are, these plasma membranes are highly semi-permeable or selective permeable, but this is definitely because of proteins present on the cell membrane of cells which allow water to pass through it, and through those. So if I put these cells in water, if I wash these cells, first of all, why do we wash? To take out some debris, some dead cells and all. But the question is, if we wash it with water, then water will go inside the cell following the law of osmosis, and cells will burst. So we have to wash it with a solution, sterile solution that has got the same osmolarity as inside the cell. And when I put some NaCl, KCl, K2HPO4, Na2HPO4, NaH2PO4 and all, so just to maintain osmolarity of the solution as well as pH is maintained, so that neither cytosolic water comes out and nor this buffer water goes inside the cell, because osmolarity is same and there will be no effective osmosis. Okay, after washing it, what we do, we will put some trypsin in it. As I said, that trypsin is an agent that dissociates cells from its substrate surface. So let me take some So I have taken around 1.3 ml of trypsin. Okay, if I can show you, and I will just put this trypsin in this flask. Okay, and then I will nicely spread it out throughout the flask. Okay, I will nicely spread it. I will just close the surface, close the bottle, the flask, And as I said that I generally give 2 minutes 2 minutes time for its function. Its function means it would be cells will be dissociated from it in just 2 minutes. Okay, and what I'll do for these 2 minutes, I will keep it in a incubator. So what is incubator? Incubator is something where so here you see that I have kept it here, and I will just close the door. So for 2 minutes, it will take 2 minutes time to get dissociated. Now this is a cabinet, this is a machine or cabinet where you have a fixed temperature. So we need to grow these cells at 37 degrees centigrade, right? And also it has some 5% of, it has some, right now it's 2.2% of CO2, but generally we need to maintain 5% CO2 concentration inside the cabinet in 37 degrees centigrade, same as our body. Okay, so even if you think of our body, then in the blood concentration of CO2 is about 5%, which is needed, which ultimately reacts with H2O and forms bicarbonate, and bicarbonate is important for maintaining a buffer environment inside our body. So this bicarbonate system is the main buffer system in our body. In the same way, here also we have to maintain this 5% CO2 so that there will be buffering. Okay, so when bicarbonate buffer system will make, it will resist the change of pH. So the reason behind maintaining 5% CO2, and maintaining means when we keep the cells, when we grow the cells, we maintain this 5% CO2 so that pH wouldn't change. And second thing, why the temperature is 37 degrees centigrade? Because that's what the temperature of our body, and that's what the temperature of the lady who is So you got my argument that even though I call that the lady is alive in my hand, but I do not completely agree with the fact that a live cell is really a definition of life. Live cell really definition of life? Because as I explained that I would call it very sophisticated machine, as good as a well-defined microscope. So when I said my eye is also a machine, but it is how does it function? Like, it has different cells. In my eyes there are different cells, and these different cells have different protein worker, and these protein workers function to give you a particular function. Okay, so like right now I can see. In the same way, when we talk about this microscope, then this microscope is also a machine. It has different function. We have light source, we have diaphragm, we have objective, lenses, and focus point, and different things, and it also functions in a way. So what makes a cell very different than this instrument? I would say just some sophistication. Like your cell machinery is more sophisticated than this. My eye is more sophisticated than this machine. That this machine requires electricity, but I do not require electricity. Second, I am on my own. Whenever I want to close it, I can close, and when I want to open it, I will open it. I am on my own. But the question is, who am I? When I say I am on my own, who am I? So when I say I am closing my eyes, then essentially there is a thought formed in my prefrontal cortex here, and that thought, in prefrontal cortex area of our brain, there are neurons which interact with each other and gives you a property we call thought, and that neurons again fires means some current goes on by depolarization, repolarization, by some changing voltage or potential and creating some potential difference that I'll talk in some other video, because trypsin can, if I leave it for a long time, it will destroy my cells. No, so the thing is, who am I? So some thought forms here, and then that goes, and it just tells my eyes, signals the eyes that these neurons will make you do a thing. Like if I want to close my fingers, so who did that? Thought formed here, and it came here, and I could close my fingers. So because of neurons. But again, when a thought forms, from where thought comes? And is there a free will that am I choosing the thought, or thought just comes as it is? Like right now, I do not know that which hand I will open, which fingers I will open. Okay, right now I do not know. Okay, trust me. And if I can guess, then I can say this and this, no issue. Okay, I just thought and I opened it. Okay, now you'll say that based on your decision, your thinking, it got opened. But why did I get the thought that I should open this? Now, see, this is a very there's an argument that it's not this much complicated. You have to think one thing. No, you have two options. But sometimes, what about new thought? That you get a new thought, new idea, from where that comes? So, and then you say, I thought, I think, who am I? So in the same way, when I said that, I forget what I said, but in the same on the same path, when I said that, what did I say? I said about the lady. When I defined us, should we really call a cell living and a microscope non-living? What makes it a different? It's the same different protein machinery doing different work, and very sophisticated machine. I acknowledge that fact, but what makes it living? And that a non-living microscope is not living. It is also doing some work. So you should call it microscope is living, but a smaller living thing, and a cell is living, but a bigger, sophisticated living thing. I don't think so. So when I say I, it's not, I have read some arguments about it, that again I'll not talk in this lecture. Means in this talk I shouldn't call it lecture unless I get a professor degree. One of my friend called me professor. And somebody asked me that, do you want to become a professor? No, I just want to know things, and for that I don't need to become a professor. Then don't you want to become a professor? Of course I want to become a professor, but that is not my goal. My goal is just to know things, just to discuss about things, just to know, and then prove that what I got to know was wrong, and then regain a new insight. So all these things, and 2 minutes are over, more than 2 minutes I will say. Okay, so here the cells interesting. I'll see it in a microscope. Yeah, very good. I can see that cells have already dissociated, and they are floating, moving on the they have dissociated from the surface. Okay, so we will again go to the cabinet and see what can be done. Now you should be asking the question that now your cells is in trypsin, and how will you separate trypsin from how will you dissociate trypsin from the cells? So trypsin can be inactivated. You can inactivate the trypsin. As I told you, trypsin is an enzyme which is used to dissociate the cells from the substratum, and this thing can be, you know, you can inhibit the function of this enzyme we call trypsin by adding this media. So I have taken some media in it, okay, and I will put it in the inside this, what we call it, flask. And as soon as I put this media in the trypsin added flask cells, trypsin activity is lost. Now trypsin is no more functioning, okay, and trypsin is no more functioning. And then what I'll do, I'm just mixing it so that all the cells get dissociated properly. And dissociated cells, I want to collect these cells to split in other cells. But the question is, should we really take this cells and divide in those different flasks? I would say no, because these cells still have trypsin, although the trypsin is inactivated, but the trypsin is still there. Now what to do? The question is what to do. So it's very simple. So we will take the cells out from this flask. I will show you. I have taken flask out from this. I have taken cells out from this flask, and I will put it in another tube. I have put it in another tube. I have closed the. So you see, now all the cells are in it. Okay. So cells are in the media, but trypsin is also there, and I don't want to take these cells and put in another flask like this. Okay, why shouldn't we? Because trypsin is still there. So what I'll do centrifugation. So centrifugation, I'll show you what it is. So you see, you come here, there is a machine. Okay, so I will put it in here. So it has around 4.5 ml solution, and this thing is having only 4 ml of solution, and this thing is having, yeah, so I guess this is fair enough. So it has less than 5. It should be working, and this is also having no. So I think I should throw some water out of it so that it become around 4.5. So basically I will centrifuge. Centrifuge means, I'll show you. This, if somebody has not used centrifuge in their life, they must be thinking, what the hell is going on? I'll tell you this. It sounds good. Okay, so this is just a weighing balance. Okay, I'll put it here, and this is the cells in the media. It has also volume of around 4.5 ml. Okay, and put it here. This sounds good. Now I will close it. I have closed it. Okay, so now just to explain what it is, I will just open it. Okay, what happens, that this thing rotates. You see, this thing rotates, and when you rotate this at let's say 250 rpm or maybe 500 rpm, 500 times per minute for 5 minutes, then in this cells, or the solution where cells are there, cells are heavier, so they will go and settle at the bottom, and all the media will be up, and then I'll throw the media. Okay, so I will throw the media, and I'll use the cells which will settle in the bottom. Okay, and this thing is needed to put there the balance, so that there should be a balance. Okay, when it will rotate with a high speed. Sounds good? Yeah. Okay. So if you can see, it has 250 rpm for 5 minutes. Now I will just start it. Okay, now it will run for 5 minutes. So what will happen, Okay, so in these 5 minutes, what will happen? That all the cells will be settled down at the bottom because of centrifugal force. If you remember that if something is rotating in a circular motion, then there is an outward force, and that outward force we call, what do we call it? So we call it we call it you know centrifugal force, and the value of that centrifugal force is mv square by r. Okay, so now the question is now the question is now the question is what is mv square by r? So m is the mass, v is the velocity, and r is the radius. Okay, now what happens that now what happens that so I always get to think about other issue, and when I think about other things, then I lose the track, and right now I was thinking about something else. Okay, so basically when mb is square by r, when cells have definitely more mass than the media or trypsin or the glucose or everything, so mv square by r will be more, centrifugal force will be more for the cells that are inside this tube, and cells will experience more force, and they will settle more, they will go down at the bottom and they will settle there. And another thing is this, that this media thing which has got lesser, and they will experience lesser force, centrifugal force, and they will be on the top, and I'll throw them out, all the media and trypsin which are not needed. I'll take out the cells and I'll dissolve the cells in some media, and after that so after that I will dissolve the cells in some other media, and then I will split it. Now the question is, should we really call cell alive? We should leave this question right now, because if you have multiple arguments, then you can only confuse people. And right now I am having multiple arguments, so I will think more about it, and then I'll tell you. But right now there is 5 minutes waiting, and what should I be telling you? So so this is the first time I am looking for the words. The reason is I can talk a lot about what should we talk. The first thing is why to talk. Why should we talk? The answer to this question is, why shouldn't we talk? When I say that why am I making this video and uploading it on YouTube, the another argument is, why shouldn't I? So then say that one argument is people may call it show off. Now the counter argument is, why should I care about people unless I am harming them? So since I am not harming them, I can do whatever I want. Second argument is, in my understanding, do whatever you want to do. It's your life, your world, your limited time. Do what the hell you want to do. I have been with people who made me continuously think that what should I do and what should I not. And right now I'm blaming them. This is also not good. Churchill has written a book, and the name of the book is some insecurity about the world, like how insecure the world is, and are we living in post-truth era, and is this really a post-truth era that has come up in the last some few time? And people started thinking it, like wow, yaar, he coined a new term, post-truth era. Sure, people are lying, it must be post-truth era. But it has always been like this. There is always truth and lie that people have been telling. It's our intellect and it's our logical thinking that should make us believe that what is truth. We need to seek for the truth. Truth doesn't you know uh, comes up very easily. And also, so there is a thing that whatever you think based on your conditioning, based on your assumptions, based on your understanding, you see the world through that lens only. So most often, if you are a liar, you will think that whole world is a liar, and if you are a truthful person, then you will think the whole world is truthful. Now, what should a wise man do? A wise man should, or woman should, always look closely for clear understanding that what is right, what is wrong, what is truth, what is untrue, and that's how you can find the truth. Okay, so I think 5 minutes are over. I am going to take the cells and I'll show you what should we do with that. So we need to go to the cabinet, and again we are here. Okay, so you must be saying here that cells have settled. So these hazy thing, no, these are cells. Cells have settled, and the media containing trypsin is up above. Okay, I'll just make you look at this angle so that you understand things properly. I don't know if I have messed up, the orientation of my camera phone.